photosynthesis in higher plants takes place in two phases, which was proposed by Blackman. According to Blackman, the photosynthesis occurs in two phases called as light reaction and a dark reaction. In the first phase, light is used and water is split to produce oxygen. In the second phase, light is not necessary, hence it is called as a dark reaction, and carbon dioxide is reduced to glucose. Then we have studied the light reaction, which includes the photophosphorylation, where ATP is produced, and that ATP is necessary in the dark reaction, and also during the non-cyclic photophosphorylation, also called as Z scheme electron transport, where NADPH2 is produced. So this NADPH2 is necessary in the dark reaction. So that is why if light reaction does not take place, dark reaction will never occur, because ATP and NADPH2 are essential in the dark reaction. So hence, especially the non-cyclic photophosphorylation is required or essential for the performance of the dark reaction. And light reaction takes place in the thylakoids, in the grana, where chlorophyll, that is the photosystems, photosystem 1 and photosystem 2. So the PS1 and PS2 are present in the grana, that is the thylakoids, and hence it takes place in that region. But the dark reaction does not require light, hence it occurs in the stroma, that is the matrix of the chloroplast, where the PS1 and PS2 are absent, and also light is not necessary for the dark reaction. So dark reaction is called as C3 pathway because the first stable compound formed is the 3-carbon compound, that is phosphoglyceric acid, hence it is called as C3 pathway. So RuBP combines with the CO2, and this CO2 when it combines, RuBP is a 5-carbon sugar, so it forms a 6-carbon compound which is unstable, and it soon splits into 2 molecules of 3-carbon compound, which is a stable compound called as phosphoglyceric acid. So the first stable compound formed in the dark reaction is 3-carbon compound, hence it is called as C3 pathway. And this pathway was discovered by, that is the intermediates of the dark reaction were discovered by Melvin Kelvin and his associates, and hence the dark reaction is called as Kelvin cycle. So it is called as cycle because RuBP, which is the starting compound, it is regenerated in the cycle. So RuBP produces 2 PGA. Similarly, 6 RuBP and 6 CO2 enter the cycle and enter the reaction and form 12 PGA. Now this 12 PGA is reduced by NADPH2. So and it forms phosphoglyceraldehyde. So out of the 12 PGALD, so 12 NADPH2 are necessary to reduce 12 PGA into 12 PGALD, so phosphoglyceraldehyde. So 2 PGALD they combine to form a molecule of glucose, whereas the remaining 10 molecules, they involve in the regeneration of the 6 molecules of RuBP. So that is how the Kelvin cycle takes place, and where this C3 pathway takes place, those plants are called as C3 plants. But there is one more cycle which is called as the C4 pathway, or sometimes it is called as Hatch-Slack cycle. So Hatch-Slack cycle, or it is also referred as the C4 pathway. Now this is an alternate pathway used in the carbon dioxide fixation where the 4 carbon compound is the first stable compound. So C3 pathway we call it so because the first stable compound is a 3 carbon compound. Now when the first stable compound formed is a 4 carbon compound, then we call it as C4 pathway. It is called as C4 pathway. So in case of maize, jawar, sugarcane, amaranthus, artiplex and many others. So there the C4 pathway takes place. So the C4 plants, they have a characteristic anatomy, leaf anatomy, which is called as the Kranz anatomy. Now why these plants like maize, then sugarcane and even this jawar, maize, sugarcane, jawar, amaranthus and artiplex, they conduct C4 pathway. C4 pathway is not present in all plants. Only in some plants this C4 pathway is present. There is also C3 pathway, but other than this C3, this C4 pathway also takes place in maize, jawar, sugarcane, etc. Now this these plants, they are having a characteristic anatomy in their leaves. So this anatomy is called as Kranz anatomy. So Kranz anatomy, what is this Kranz anatomy? So the leaf anatomy, which is having a characteristic, here the bundle sheath cells surrounding the xylem and phloem. So these bundle sheath cells, they are large, and when they are large, the bundle sheath cells, they contain also larger chloroplasts. If the cells are large, the chloroplasts also are large, and they don't contain grana. So the chloroplasts of these cells, they are not having grana, but they contain chlorophyll. See the difference between the chloroplasts here. So usually when we say chloroplast, we mention about grana, thylakoids and everything. But the chloroplasts of the bundle sheath cells, which are large and contain also larger chloroplasts, but without grana, but they contain some starch grains. So starch grains are present in these chloroplasts, but no grana. But in the neighboring mesophyll cells, it is quite opposite. The chloroplasts are smaller and grana are present, but no starch grains. So almost opposite characteristics. So in the bundle sheath cells, chloroplasts are larger, no grana, but starch grains present. In the mesophyll cells, the chloroplasts are smaller and they contain grana and there are no starch grains. So this is the characteristic which we see in the mesophyll cells. So in the these C4 plants, how this C4 pathway takes place. So there are two carboxylation reactions. So there is also C3 pathway where CO2 is converted into glucose, and also there is C4 pathway which also takes place in this cells. Now this Kranz anatomy, what is the meaning of Kranz? Actually Kranz is a Greek word which means wreath. So wreath is a type of garland which is used to honor the persons in their memory. So that means which persons who have sacrificed and dedicated their lives for the country or like that. So usually this wreath is used. It is a round ring shaped garland. We might have seen whenever the high dignitaries like the president or prime minister or any other such individuals, when they show some respect to the persons who have sacrificed their lives, so they use this ring like a garland, and they will place in the front of their memorial, and there that is called as wreath. Here the bundle sheath cells of the leaf, the cells which are surrounding the xylem and phloem, so those bundle sheath cells, they appear like a wreath. Hence, and also they are larger in size. They are different from the mesophyll cells, and that is why that is called as Cran's anatomy. So this Cran's anatomy is the characteristic feature of the C4 plants. So plants which conduct C4 pathway, and like maize, jawar, sugar cane, etc. Now this C4 pathway, also called as the Hatch-Slack cycle, which was explained by Hatch and Slack. So this Hatch-Slack cycle, how it takes place? It occurs in 4 steps. That is how it occurs. Here what happens? Phosphoenol pyruvate is a compound. Now in the these bundle sheath cells, the reaction will start. The phosphoenol pyruvate, it combines with the CO2. Now there in the Kelvin cycle, RuBp combines with CO2 and forms PGA. Here PEP, phosphoenol pyruvate, a 3-carbon compound. So this pyruvate is a 3-carbon compound. To this phosphate is attached. Phosphoenol pyruvate combines with CO2, and this forms a 4-carbon compound. So this is a 3-carbon compound, and this one attaches and forms a compound called as oxaloacetic acid. So oxaloacetic acid is formed. So oxaloacetic acid is a 4-carbon compound. Now oxaloacetic acid, when it is formed, this oxaloacetic acid, it takes place in the presence of an enzyme called as PEP carboxylase. That is an enzyme, like how the Rubisco involves in Kelvin cycle in the first step, where RuBp combines with carbon dioxide. Here also PEP, phosphoenol pyruvate, combines with CO2 in the presence of an enzyme called as phosphoenol pyruvate carboxylase, and a 4-carbon compound like oxaloacetic is formed. Now this oxaloacetic acid is reduced. So oxaloacetic acid, which is produced here, it is reduced to aspartic acid or malic acid. Malic acid, or sometimes it is also formed like aspartic acid. So there is reduction. So it is reduced by the electrons. And then here malic acid is also a 4-carbon compound, Aspartic acid is also a 4 carbon compound. So oxaloacetic acid either forms malic acid or aspartic acid. So in the next step what happens, this malic acid which is formed, now it is decarboxylated to form a compound called as pyruvate plus CO2. So pyruvate is formed, which is a 3 carbon compound, and this is a 4 carbon compound. So 1 carbon is lost in the form of carbon dioxide, and pyruvate is formed. Now this pyruvate will go to the mesophyll cells. So this takes place in the bundle sheath cells. It occurs in the bundle sheath cells. Then what happens? The pyruvate will enter into the mesophyll, and this CO2 which is released, it helps in the Kelvin's cycle. It enters into the Kelvin's cycle in the mesophyll cells. Then what happens in the last step? So this pyruvate will regenerate to form PEP. So it is sent back to the mesophyll cells, and where PEP is regenerated from pyruvate in the presence of ATP. So using ATP, this phosphate will attach to the pyruvate to form phosphoenol pyruvate. Again pyruvate is regenerated. So the starting compound is regenerated, and hence that is called as the cycle. So when the starting compound gets regenerated, it is a cycle. The Kelvin cycle is also called because RUBP is regenerated. This is C4 pathway. And the next aspect of this is photorespiration. What is photorespiration? Next aspect. Now photorespiration is actually related to photosynthesis, to dark reaction. However, the word respiration is used here. Photo, light. Respiration in presence of light, that gives the meaning. But here we refer it as respiration because oxygen involves in this process. Now in the photosynthesis, so far what we have studied, during light reaction water is split and oxygen is released. Oxygen is released through the stomata. In the dark reaction, CO2 is reduced, or CO2 is used to form glucose, that is in the dark reaction. Now in case if oxygen accumulates in the cells, oxygen is not a poisonous substance. If oxygen accumulates in the cells, usually during light reaction it is liberated into the atmosphere through the stomata. In case if it accumulates in the leaf cells, here it will inhibit the dark reaction. What happens when the Rubisco enzyme gets oxygenated, means in the presence of oxygen, so the PGA will not be formed. So when there is no formation of PGA, automatically the further PGA will not be converted into PGLD, or there is no formation of PGLD, no formation of glucose. So what happens? RUBP and CO2, when Rubisco is oxygenated in the presence of oxygen, it will not form PGA. Instead, it will form a compound called as phosphoglycolate. That is the compound formed. Or sometimes it also formed phosphoglycerate. So these compounds are formed when the oxygen is present in the cells, when the dark reaction is going on. So this phosphoglycolate or phosphoglycerate, which are formed by the presence of oxygen. So when there is no PGA, no question of formation of glucose. So this process is called as photorespiration. So this is called as photorespiration. Photorespiration means it is a process where the enzyme rubisco gets oxygenated and the RUBP causing some of the energy produced by photosynthesis to be wasted. So here the energy which is formed during the photosynthesis, it is wasted there. So that is why photorespiration is a wasteful pathway. So when glucose is produced, it is useful, and the oxygen which is produced, it is also not useful for respiration to the animals. It is not released outside. That is why it is a wasteful pathway, and that actually competes with the Kelvin's cycle. Then this process occurs in chloroplasts, peroxisomes and mitochondria. So that is the location or the site of the cells where the photorespiration takes place. So the RUBP, instead of converting into PGA, it will bind with the oxygen. So the RUBP will combine with the oxygen where the enzyme also will be oxygenated and form phosphoglycerate or sometimes phosphoglycolate. So there is no synthesis of ATP, and there is no synthesis of sugar, and also NADPH2 is also not reduced. So that is the photorespiration. So it results in the release of carbon dioxide with the utilization of ATP, and hence photorespiration is a wasteful pathway. Then lastly, that is one short aspect related to the photosynthesis. Lastly, what are the factors affecting photosynthesis? So there are different factors. There are internal factors which are called as the plant factors. There are external factors. So various factors are there that affect the rate of photosynthesis. Now inside the plant there are some factors which are called as the internal factors. Now these internal factors, they may be the number of the leaves in the plant, the size of the leaves in the plant, the age of the leaves, and the orientation of the leaves, and the mesophyll cells in the leaves, the chloroplasts, and the CO2 concentration in the cells, and the amount of chlorophyll. See, in a plant, if there are more number of leaves, naturally we can say, if there are more number of leaves, more amount of photosynthesis. Definitely, naturally. And if the size of the leaves are small, there is only little amount of photosynthesis. If the leaf size is large, there is more photosynthesis. And even the age of the leaf also is important. If the leaf remains for a long period, there will be more supply of the food to the plant for its development. If the leaf falls within a short period, if the age is very less, automatically the plant will get less food, less amount of food material. That is how the age of the leaf is very important. Number of the leaves important, size of the leaves important, means that affects the rate of photosynthesis, and also the age, age of the leaf. So number, size, age, and orientation of the leaves. How the leaves are orientation. If the leaves are having a direct, receiving direct sunlight, they will get a sufficient amount of solar energy. If the leaves are not inclined to the light, So that means if the rays are passing in a particular angle, if the leaves are along that rays, so the rays will not fall on the leaves. They will not be properly received by the leaves. Orientation of the leaves is also very important. And also the number of the mesophyll cells in the leaves, that is also important. Then the amount of the CO2 concentration which is necessary, so that CO2 which enters into the leaves, that internal CO2 present in the plant, it is also having an effect on the rate of photosynthesis, plus the amount of chlorophyll is also important. So leaf may be large. If suppose if there is a small leaf, if there is a large leaf, maybe in same plant or different plants. So when that is the case, if the leaf is small and large, we believe that the large leaf is having large amount of chlorophyll. This is having little amount. But in the large leaf, if the amount of chlorophyll is equal to the small leaf, the photosynthesis will be same. So the size will not be affected. Size is not taken into consideration. That is why the amount of the chlorophyll is also important. Means these are the internal factors, also called as the plant factors. Now if you come to the external factors, for example light. Light is one important factor. We know that light is necessary for photosynthesis. So light factor. Then there is temperature. So light, temperature is another factor. Then CO2 concentration. Now CO2 concentration means in the atmosphere. It is also considered as internal factor. So it is the internal CO2 concentration, the amount of CO2 available inside the leaves, that is important. Now the CO2 concentration of the environment, which is normally 0.03%. So that is how it is affecting. Then water is also another important factor. So like that, these are the environmental factors: light, temperature, CO2 concentration, water, which affects the photosynthesis. Now one by one, how these will affect the photosynthesis, we shall see. Now light. So the light intensity, the light quality, the light duration is important. Intensity of the light, how much intensity of the light is necessary. So now when we take the unit of foot candles, about 2500 foot candles of light intensity is necessary for photosynthesis. Now early in the morning there is sunlight, in the afternoon also there is sunlight, but that sunlight is different because of the intensity. In the morning the intensity is very low, though sunlight is received, and in the noon there is sunlight, but it is very having high intensity. So that is why the intensity is very important in light. So very high intensities also will not increase the photosynthesis, and very high intensity will cause bleaching of the chlorophyll molecule. So that bleaching of the chlorophyll molecule occurs which does not involve in the photosynthesis, that automatically the photosynthesis will get reduced. And however, under low intensities also there is less photosynthesis. That means early in the morning when light falls on the leaves, that light is not sufficient for photosynthesis, because light is necessary for photoexcitation, for excitation of the electrons in PS1 and PS2 to conduct cyclic and non-cyclic photophosphorylation. So that is why low intensities cannot cause excitation, and thus there will be no cyclic and non-cyclic overall the photosynthesis will be reduced. So in light quality, what wavelength is received by the light? So when the light is having 7 wavelengths, if that light is not having red and blue wavelengths, automatically the photosynthesis will get reduced. So this photosynthesis is reduced because of the wavelength. So it is the quality of the light which is very important. So that what wavelength is present in that light which is received by the plant. Then the duration, how much hours the plant has to receive light. Of course, when we go to the duration, it is a long process here. There is a phenomenon which is called as photoperiodism, where the plants require minimum and maximum amount of light, the duration of light, and that that varies from species to species. Some require less duration of light, some require more duration, more hours of light. Like that, the duration is very important. However, low intensities for long duration will help in the photosynthesis. So though there is low intensity, but however for long duration it will favor the photosynthesis. That is about light duration. Then coming to temperature. So temperature is very important. So temperature, optimum temperature is necessary for the functioning of enzymes. Now in the dark reaction, we just mentioned about Rubisco. If there is no Rubisco, or if the Rubisco is inactive, RUBP will not combine with CO2 in the absence of the Rubisco. So Rubisco is active only under optimum temperature. So temperature is important for the plants, and especially this temperature differs from plants to plants, from area, zone. For example, we refer as temperate regions and tropic regions. Temperate means there are regions which are cold, somewhat low temperatures. Tropical means they are somewhat hot. So plants living in tropical means tropical plants and temperate plants, they are having different optimum temperature. So there is no fixed optimum temperature for all plants. So that is why temperature helps in controlling the activity of enzymes, particularly in the dark reactions. Now however, C4 plants, they respond to higher temperatures, while C3 plants they respond to low temperatures. So C4 plants, even if there is high temperature, they will respond to that, and the C3 plants, they also respond to the low temperature. This is how the these tropical plants, they require higher optimum temperature, whereas the temperate plants, they require low optimum temperature. Then if you come to the CO2 concentration, already I mentioned in the atmosphere there is 0.03%, which is sufficient for photosynthesis. If the concentration CO2 or the proportion of the CO2 falls, definitely the photosynthesis will decrease. But however, if the CO2 increases, so which is 0.03%, it will increase only up to 0.05%, up till this. If the CO2 increases up to 0.05%, then there is increase in CO2 fixation. But beyond this, there is damaging over long periods. So that means excess of CO2 above this concentration will be converted into carbonic acid, and the cells become acidic, and ultimately the enzymes will not function, and thus gradually, if the increase the CO2 is there, it will cause damage to the plant for long periods. But C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis. That is the effect of CO2 concentration. And ultimately water Water, we know that it is necessary in the light reaction. As we had seen in the light reaction, water splits to produce hydrogen ions or protons and helps in the formation of oxygen. We call it as the photolysis or photoionization of the water, and thus water is essential for light reaction. If there is no water, no production of oxygen, so the PS2, which has liberated the electrons, will not be brought back to the normal state. And also water helps in the opening of the stomata. The stomata, which is having a pair of guard cells, these guard cells, they open only due to the presence of water by maintaining the turgidity of the cells. If there is water stress or if there is water scarcity, they lose, the turgidity is lost, and ultimately the stomata will close, and thus the CO2 will not enter into the cells, so it affects the or it reduces the availability of the CO2 in the leaf cells. And also, if there is no water, the leaves will wilt. That means the leaves will fade, in other words, and thus the leaf surface area, that means when they wilt, the leaf surface area is also reduced, and thus overall the photosynthesis gets reduced. That is the effect of water. And lastly, there is one law which is called as Blackmans Law of limiting factors. This is a law which is related to the factors itself. We know that photosynthesis is controlled by various factors. So light is necessary, temperature is necessary, CO2 is necessary, water is necessary, like that. So what Blackman proposed, one law called as law of limiting factors. Now actually before Blackman, there was one law which is proposed by Liebig's law. Liebig's law of minimum, it is called as Liebig's law of minimum. Now this Liebig's law was slightly modified. It was modified by Blackman. And what is that Liebig's law? We are not concerned. So this Blackman's law, what it states? Now in photosynthesis, we know that it is controlled by several factors: light, temperature, CO2, water, chlorophyll, and all that, isn't it? So CO2, water is necessary, light is necessary, temperature is necessary, even chlorophyll is also necessary, isn't it? Now Blackman's law states in general, but here we are referring with reference to photosynthesis. Now here you see all these factors are important. Light is important, temperature is important, CO2 is important, water is important. There is no any equal, means one is more important, one is less important, like that. All are equally important. If any one of the factor is not there in proper concentration, photosynthesis will not take place. Light, when there is no proper light, no photosynthesis. No temperature, no photosynthesis. No CO2, no photosynthesis. No water, no photosynthesis. Like that, every factor which involves in that process is controlling that reaction. If among all the factors, if you control or if you minimize any one of the factor, the process is controlled. So whenever a physiological process is controlled by several factors, one of the factors will govern the rate of the reaction. This is what Blackman explained. So he states that when a process, of course we are referring this with to photosynthesis, but Blackman's law is concerned to all physiological processes. So when a physiological process is conditioned as to its rapidity by a number of separate factors, so when a physiological process is conditioned as to its rapidity by a number of factors, separate factors, the rate of the process is limited by the pace of the slowest factor. So which factor is the slowest, that factor will control or will limit the rate of the reaction. That is why limiting factors limit. All factors are capable of limiting the rate of the reaction. So law of limiting factors. So each factor has the capacity of limiting, has the capacity of limitation, and thus the physiological process, this is what the Blackmans law of limiting factors explain. So the law of limiting factors state that when a physiological process is conditioned as to its rapidity of factors which are separate as to its rapidity of separate factors, the rate of the process is governed by or is limited by the process of the slowest factor. So which is the slowest factor? So that factor will limit the reaction. So every factor has the capacity of limiting the reaction, law of limiting factors. This is proposed by F.F. Blackman, who also said that photosynthesis occurs in 2 steps. Initially we mentioned. So photosynthesis occurs in 2 steps, 2 phases, light reaction and a dark reaction. So he proposed this law of limiting factors by modifying the Liebig's law of minimum. So that is about the photosynthetic reaction and the various events in the light reaction, dark reaction, the C3 pathway, and also the C4 pathway called as the Hatch-Slack cycle. Then what is photorespiration, then factors affecting the photosynthesis like light, temperature, CO2 and water.